Hello everyone, this is Sheethal Maria with your BioOcean, and today we are here to learn one of the classic topics of biology, that is DNA, deoxyribonucleic acid. So if any one of you are new here, just go to my channel and subscribe it and press the bell icon so that you can get the notifications whenever I upload the video. And let's get into the video. Talking about DNA, we can say that it is almost same in majority of the organisms, and all our cell is composed of a DNA in its nucleus. So only few exceptions are there. In case of some animals, the genetic material is not DNA, but still majority of the organisms are having DNA as their genetic material. And we can say that the DNA is composed of mainly three items, that is a pentose sugar, a phosphate group, and nitrogen bases. As we can see in this picture, the phosphate group is shown in orange. The sugar here is deoxyribose sugar, that is shown in blue, and the bases, nitrogen bases, are shown in green. Basically, a DNA has two types of nitrogen bases, purine bases and pyrimidine bases. Purine bases are of two types, adenine and guanine, and pyrimidine bases are of two types, thymine and cytosine. This adenine can only bind to thymine with 3 hydrogen bonds in between, and the guanine can bind only with cytosine with 2 hydrogen bonds in between. No more kind of pairings are possible in these 4 nitrogen bases. It was actually Edwin Chargaff and his co-workers in 1950 who made major conclusions about the base composition of DNA. You can see Edwin Chargaff in this picture, and all his conclusions were made as a rule named as Chargaff rule, and it states that the sum of purines is equal to the pyrimidines, that is equal to 1, and the ratio of adenine to thymine is 1. Also, the ratio of guanine to cytosine is also 1. That is, the amount of adenine is equal to the amount of thymine, and the amount of guanine is equal to the amount of cytosine. For simplifying this, we can see a picture. Here it shows that A is equal to T and G is equal to C. Okay, therefore we can say that purines is equal to pyrimidines, and altogether it creates 1. So that we can say that the DNA is symmetrical. One drawback that his studies possessed was he could not get up into a conclusion about what is the structure of DNA is. So it made a lot of controversies and all those for the acceptance of this rule and stuffs. And later, several forms of DNA was described, like A form, B form, C form, Z form, like that. And the most common one among these is the B form, which was described by Watson and Crick. Watson and Crick together produced this model and named it as double helical structure of DNA. And this picture shows these two scientists, Watson and Crick. The salient features of this model is that the DNA molecule consists of 2 polynucleotide chains which are coiled around a common axis in the form of a right-handed double helix. We can assume this as a helical staircase wound in a right-handed fashion. And the phosphate and deoxyribose units were found on the periphery, whereas purines and pyrimidine bases occur in the center. We can say that this sugar, deoxyribose sugar, and the phosphate together created the backbone for these nitrogen bases to exist in the center of them. Okay. Then the diameter of the helix is 20 angstrom, and these bases, nitrogen bases, are 3.4 angstroms apart. Therefore, helical structure repeats after 10 residues on each chain, that is at intervals of 34 angstrom. See this picture, the diameter is shown as 20 angstrom, and one completed turn with 10 bases comes after 34 angstrom. After that, it takes one loop, and we can say that this picture shows the DNA is wounded in a right-handed fashion around a common axis, around a central axis. This is the overall structure of DNA. The two chains are held together by hydrogen bonds between the pair of bases. I have already told this, that adenine and thymine have 3 hydrogen bonds in between, and guanine and cytosine have 2 hydrogen bonds in between. That is which makes them come closer, and this DNA molecule resembles a winding staircase with sugar phosphate molecules of the nucleotides forming the railings and linked together nitrogen base pairs forming the steps. Suppose you are having a staircase which is helical and in a right-handed fashion and resembling to the DNA, and this staircase have railings which is similar to that of phosphate and the sugar of DNA, as well as the steps in between, which resembles the nitrogen bases in the DNA. Okay, this is what the resemblance in a staircase and a DNA are. The polynucleotide chains are antiparallel, that is, they have complement base pairing. Antiparallel means if one chain starts from 5' and ends in 3', the next chain will be starting in 3' and ending in 5', and in between the nitrogen bases. In case of nitrogen bases, if one strand has a G in it, opposite to that in the second strand there will be C. Like that it goes on. That is how it comes as complementary pairs. Okay, you can see that in this picture. If in one strand T comes, opposite to it will be A. If in one strand C come, opposite to it will be G. Like that it goes on. This is how it becomes anti-parallel as well as complementary. This is the whole structure of DNA. Hope you understand it. If you like this video, give me a thumbs up, comment your suggestions, share it to your friends and all those who are interested to learn biology, as well as if you have any kind of doubts, let me know through my email ID that I have provided. And let's see you in the next video. Thank you for listening. God bless. Thank you.